Then the vasa recta, it also helps to maintain concentration gradient in renal medulla by preventing the washout of solute, both water and sodium chloride. Next, as a blood flows through the descending vasa recta, it picks up a solute from interstitial fluid and become more concentrated. Once the fluid exchanges occur in interstitial fluid, and again now it is picked up from or drain into from interstitial fluid, so that it is more concentrated, and blood flows through the ascending vasa recta. It releases the solute into the interstitial fluid, which helps to maintain high concentration solute in medulla. And the countercurrent multiplier and vasa recta are essential mechanism to place kidney ability to concentrate in urine. So countercurrent, which involving loop of Henle and collecting duct, establish the concentration gradient in medulla. Main thing. Vasa recta is current exchange the blood supply in renal medulla and helps to maintain gradient. So together mechanism to ensure the kidney can effectively concentrate urine to maintain the body's fluid and electrolytic balance. Here the type of nephrons. Types of nephrons are cortical nephrons and juxtamedullary nephrons. What is cortical nephrons? Here the cortical nephrons are abundant type comprising approximately in 85% of all nephrons. They are primarily located in outer region of kidney cortex. Okay. Outer region of kidney cortex have shorter loop of Henle. These nephrons are involved in maintaining overall body fluid balancing. Then juxtamedullary nephron. Here the juxtamedullary nephrons are located closer to the medulla of kidney and have longer loop of Henle. So these also play an important role to producing concentration of urine. Okay, and involved body water conservation mechanism. Types of nephrons are cortical nephron and juxtamedullary nephron. And see, this is the cortical nephron and juxtamedullary nephron, which is important for playing concentration in urine. Conclusion. Here the conclusions are: nephrons are the functional unit of kidney, vital task to maintain our body fluid balance and regulate the blood pressure, eliminate waste product from the body, which integrate a system, ensure the proper filtration, reabsorption, secretion, and the process required for all over health well-being. Understanding the structure and functions of nephron, the types providing very valuable insight to complex procedure involving urine formation. Here the nephrons are remarkable capabilities enable to body to maintain the delicate balance and eliminate the waste effectively. And there are two assessments are presented here, which is iron present in both secreted and absorbed by nephron, and second part is part of nephron is impermeable to water. Here the answers are ions that are secreted and absorbed by nephron is potassium ions, K+. It is important for maintaining proper electrolytic balance in the body, and it is level or tightly regulated by nephron. And second one is the part of nephron is impermeable to water are ascending limb of loop in Henle is impermeable to water, and the section of nephron plays a crucial role in establishing concentration gradient necessary for water reabsorption in collecting tube. Here the fate of sodium and water in the renal tubules are step 1, the active NaCl reabsorption without a water. The thick descending limb make a tubular fluid and more dilution. So the interstitium is concentrated. And step 2 are the fluid in descending limb equilibrate osmolarity. The hydrostatic interstitium by the movement of water out of the tubule. Again the step 3 are concentrated fluid in a descending limb flows a counter current which is ascending limb where the NaCl is passively reabsorbed. So further concentrating in the medullary interstitiums are equally possible, and as well as water in the reabsorbed corticals and outer medullary collections are urea concentration and tubular fluids rises markedly. So the urea then moves down passively out of the inner medullary collecting duct, then into the interstitium. So vasopressin and regulated urea transport the urea contribute to the increasing interstitial osmolarity. And step 5 are vasa recta have a high heparin configuration. Similarly to the loops of Henle, it is close proximally to the tubule and plays key role in maintenance of medullary osmolarity gradient. So the retaining of NaCl and water is reabsorbed and systemic circulation is maintained. Thank you.